All right, hey there. So we are going to be doing gram staining today. In order to do gram staining, we have to create or prepare bacterial culture slides. So I have just a regular old microscope glass slide here. I have used a grease pencil to label it so we know that it's E. coli that we're adding here. And then I added just a circle of grease, again, just to sort of know that we are going to be adding our bacteria to this location. And then as we add our reagents to this, I know exactly where to add the reagents so we don't have to flood the entire slide. We could just use this specific region. It also tells us when we're using the microscope where we need to focus on the microscope. So first I need to be taking my bacterial culture. And so these are the plates that we had set up the other day. And we did the streak plate method. And so the idea, and this is a little hard to see, but the idea is that we have one very concentrated region in quadrant 1. And as we go to the next quadrants, we get less and less bacteria, and so that you can actually get individual isolated colonies. So we want isolated colonies, individual colonies that are not connected to others. Basically, we know that that started off as one bacterial cell. It went binary fission, underwent binary fission over and over and over. And now we can just isolate one specific colony. We know there's a lot of bacteria, and all those bacteria originated from one cell, which was the type of bacterial cell that we want to use. So we have Bacillus megaterium, and then we also have E. coli. Now, I'm going to be isolating a single colony that's well separated out from others. So in order to do that, I need to make sure I'm using my sterile area. So I have my Bunsen burner set up here. And I'm just going to kind of focus on the lab bench so you can see what we're doing. All right, and the lighting's not great, but it should work. So I'm going to light my Bunsen burner. So I've got my striker ready. And there we go, Bunsen burner is lit. We've got a nice blue flame, nice triangular blue flame. And we're going to do two different ways of creating our plates. So I've got one slide all ready to go. The next slide, and I'm just creating a circular region using the grease pencil so that I know exactly where to add my bacteria. We're going to be using a loop, just like we had in the past, to spread our bacteria, except I don't want to just go straight from this plate right onto this slide, because the bacteria can be kind of a little bit sticky sometimes, and really there's nothing that I'm putting them onto. So you actually want to have them go into a little bit of a suspension, so we're just going to use regular old water. Now you can use a disposable pipette to do this. However, pipettes typically give you a little bit more water than you want. So we're going to be using a micropipette today. So this is going to measure in microliters. So I'm actually going to, right now it's set at 30 microliters. You can see on the side. And then you turn this little dial to basically change the setting. I'm going to go down to 20 microliters. There is a disposable tip at the end here. And then if you push down the plunger, there's actually two stops. There's one stop, and then you can actually go a little farther. So when you are sucking up the water, you go to the first plunger, one, go back a little bit. So just down one, suck up the water, and now you can see the water, it's about up to here. Now when you're dispelling water, you go to the one plunger, but then sometimes you can see there's just a little bit left in the very tip of that. So you always want to use that second push and then actually kind of guide it along the edge if possible. That'll just get that little extra bit out. So we're going to add just a little bit of water to the central region of each of these slides, and that'll give us a little bit of liquid to basically sort of wash these bacteria off onto. Otherwise they just might stay sticky and they don't really spread around very well, and it's not that great of a slide. All right, so we're going to be using this plate, and then I also have an agar slant here, so we'll be using two different methods, or two different growth methods. So as always, if I'm using a loop, I always want to make sure I sterilize my loop beforehand, so I always flame the loop. And then let it cool for a little bit. You never want to be too hasty when you're letting this cool. You don't want to singe your bacteria. That would be very bad, so we're going to avoid doing that. And I'm actually going to use, I'm not going to use that slide, I'm going to use this one over here because we're using, this plate is Bacillus megaterium, the other slant is going to be E. coli. So I have single E. colonies, so I'm going to just scoop up a single colony. If you hear a singe, it means this was too hot. You probably just killed all the bacteria, and we're going to kill them anyways, but we don't want to singe them. And then I'm just going to spread this around that little circle of water on there. And then we're just going to put this off to the side to air dry. After we air dry it, then we are going to heat treat it. And that basically makes it a little bit more permanent on the slide so that when we add all our reagents, they don't just wash right off. It doesn't wash off our bacteria. So I'm going to flame my loop really well, kill off the bacteria that's on there. So now that slide is done. I'm going to do my second slide, which is E. coli. I want to make sure that my loop is nice and cooled before I touch my bacterial cells, because again, it will end up destroying the cells. All right, now here is my E. coli, Escherichia coli, and I've got colonies on here as well. So anytime you open up a tube, you always want to flame the top of the tube, just in case there's a little bacteria that get caught there. I'm going to go straight in now that my loop is cooled enough. And I'm just going to drag my loop straight out, and in the process, it's picking up that bacterial cells. Flame the top of the tube again, put the cap back on. And now I'm going to just drag this around that slide, and I'm basically dispelling some of these E. coli cells that I just picked up, and basically just kind of getting them in that water so that they sort of cover this little section of the slide. So that's what we are going to be staining. Always flame the loop every time you're done with it. Flame, flame, flame. You don't want to put this loop back in its storage location with a bunch of E. coli on it, a bunch of bacterial cells on it. All right, and now I put this down on the bench, which I don't want to assume is sterile anymore now that it's touched it, but I'm not going to be using that loop anymore, so it's just going to go back into storage. So now my two slides are ready to go. At least they're ready to be heat treated. It's going to take a couple minutes for that water to evaporate enough to be able to heat treat my slides. When I do, I'm just going to pass them fairly quickly through the flame, and I'll show you that in just a second.